We'll continue now the talks about biosimilars, and as Paul mentioned already, I will give a talk about the clinical assessment of biosimilars. So this illustration you may know, and it was shown already several times. And you see that there is a big basement for in vitro studies, in vivo studies, so PK and PD data. And then we have the other way around when we compare it to reference products and the clinical trials for reference products, that we have on top just a few, maybe just one clinical trial to show safety, efficacy, and immunogenicity in comparison to the reference product. So all in all, the comparison and the characterization of the reference product and biosimilar is done in very details. So it's about the primary structure, impurities, biological activity, and so on. And there are many, many tests. And all in all, I think we have more than 100 quality attributes that can be characterized with these analytical methods. So this is the main part of a biosimilar development. And this is just an example to show you the physicochemical similarity in peptide mapping, for example, that there are no differences. And this is what Paul said when he said there's no doubt that a biosimilar is similar and the characteristics are similar or the same. But in the end, we don't know the clinical outcome because we don't have a long follow-up of clinical trials. So therefore, in this stepwise demonstration of clinical equivalence showing biosimilarity between PK studies and PT parameters, and then biosimilarity between PD parameters and efficacy studies, I will focus on efficacy studies on clinical trials. And there are differences when you think of an innovative product, a reference product, and a biosimilar candidate. Usually, if you have a new innovative product, then of course you want to show superiority versus the standard of care. In biosimilars, you want to show equivalence. You don't want to show non-inferiority. You want to show equivalence because you want to show biosimilarity. The patient population for the innovative product is, you can use any. So if you want to have your approved indication for metastatic patient population, then you go with your clinical trial in metastatic population. If you want to have it in neoadjuvant or adjuvant, or let's say more distinguished subgroups, then you have to do your study in this distinct subgroup. And for the biosimilar candidate, this is recommended by FDA and EMA, you have to have a sensitive and usually homogeneous subgroup. Then we can talk about study endpoints. Of course, me as a therapist and a physician, I'm interested in the outcome of my patients. I'm interested in overall survival, disease-free survival, progression-free survival, and so on. So in survival data. And for the biosimilar, we have sensitive endpoints recommended by the EMA, having objective response rate or pathological complete remission. So it has, in fact, nothing to do with the outcome of my patients. Because if you have, let's say, a lymphoma patient, you know, after 5 years, was my therapy successful or not? And if you have a patient having 4 liver metastases and these are reduced by a treatment down to 2, then you don't know, does my patient live longer with just 2 metastases? So this is the dilemma between response rate and, on the other hand, clinical outcome and in terms of clinical benefit and survival. And then you have the issue of safety, immunogenicity, you have to observe the whole development process, and then extrapolation. And extrapolation is the talk of Paul coming after my talk. So what is a sensitive patient population? It's an immunocompetent population. That means a non-pretreated patient population, because a pretreated patient population is influenced by the treatment lines before. You have to consider comorbidities. So don't take very ill patients, patients with multiple diseases, patients with many concomitant medications, and the severity of the disease does also matter. Regarding sensitive endpoints, you have to have clinical relevant, readily accessible, and able to detect differences, because you want to show biosimilarity. The ESMO published a position paper last year, and they refer to every recommendation, and they say a biosimilar product should be tested in the most sensitive population, and we want to clarify now what is the most sensitive patient population. The regulatory view of endpoints, because we talked about endpoints already, says that progression-free survival and overall survival, as I said, is influenced by the patient itself, by the treatment lines, by the treatment duration, by comorbidities. So therefore, they don't recommend survival endpoints as sensitive endpoints. And this is a discussion that is intensively done in Germany at the moment, because we know about the dilemma I told you between response and clinical survival outcome in the end. Okay, when we talk about clinical studies, then you are aware of that we only talk about a sample. And then the indication we want to approve our product has to be correlated with the sample size. And the sample is always smaller, of course, than the whole patient population. And this is a discussion we have at the moment with, between clinical trials, the sample size is there, and real-world data. So if you go to our institute in Germany for cost effectiveness, the IQWiC Institute, then they want to know, well, okay, it's nicely shown that in this indication with your distinct patient population, a benefit was shown, but what happens if you transfer these results into real-world data? Because in real world, and all of you know who conduct clinical trials and recruit patients in clinical trials, that in the trials you have your eligibility criteria and your exclusion criteria, and the corridor sometimes is very narrow. And in real world, you don't have that narrow corridor. You have patients with more diseases, more comorbidities, and so on. So the standard, as I told you already, is the superiority design. With the question, is there a clinically meaningful difference? And this is the forest plot illustrating the hazard rate and the confidence interval. And of course, it must be in favor for your innovative product. The non-inferiority design, there has to be a lower margin, a lower boundary defined. And this is an illustration for such a confidence interval showing non-inferiority. So your product you want to compare with your other product must not be inferior, and this is the clinical trial design you have to use in this indication. Equivalence design means that you have to define a lower margin and an upper margin, and your confidence interval has to be in between. And this is to show equivalence, and this is the design that is recommended to be used for the assessment, the clinical assessment of biosimilarity. So this is the gold standard in the end, and your biosimilar product is not better and not worse than the reference product. So to include this, there's superiority, non-inferiority, and equivalence study. And the equivalence study is the gold standard for evaluating a biosimilarity. Possible outcomes of equivalence studies are shown in this slide, where equivalence met. So there is the clinical endpoint met, and the other examples, there is equivalence not met, and in special cases, maybe. And we talked about 2 days ago about a new technical possibilities we have to design drugs, to compare drugs, so we have more sensitive tests than 20 years ago. So maybe if you, the owner of a pharmaceutical company, having the idea to design a biosimilar compound, then maybe you will get a bio better in the end, because you have better technical support than 20 years ago. And this is not the goal for a biosimilar compound. That has to be shown equivalence and not superiority. Of course, we have to take under consideration some statistical and clinical considerations. Statistical reasonings, we have to define the margins quite nicely. We have to have margins to be selected such that a substantial part of the treatment effect can be maintained and potential loss and long-term efficacy is mitigated, and clinical justification has to be clinical meaningful. Response endpoint need to correlate with the long-term survival outcomes, and this has to be done later on because the follow-up of these studies done in metastatic setting or neoadjuvant setting, so these are the studies we have at the moment, they don't have a longer follow-up. So in metastatic settings, patients will die in the end, and in neoadjuvant setting, you just have your endpoint receiving a PCR, and then you have to have a longer follow-up. But this is not, but we don't have this longer follow-up at the moment from the studies that were published showing biosimilarity between reference products and new biosimilar products. So this is a case example. We have, on one hand, the neoadjuvant early breast cancer setting, and the endpoint is the total PCR. That means no residual cancer burden in the breast and in the nodes. In situ carcinoma is allowed. Then after surgery, of course, you have your adjuvant setting for early breast cancer with clinical endpoints, event-free survival, disease-free survival, overall survival. And then in metastatic breast cancer, you have objective response rate, progression-free and overall survival. And as I said, neoadjuvant setting is a proper setting to show equivalence and to be not as expensive as it is designing clinical trials for reference products, and in metastatic setting as well. But in the adjuvant setting, it takes a long time. Every adjuvant study, you are aware of this, takes ages. So we have to recruit all the patients, and then you have a statistical powerful calculation how many patients are included in the trial. That means a few thousand maybe. So it takes time to recruit these few thousand patients, and then later on you have to have a follow-up of at least 5 years. So if we have an idea now making an adjuvant trial, we will get the results maybe in 8 years. So this is not feasible for testing biosimilars. And then when we read the publications, and we usually read very carefully if a clinical trial is presented in a paper, then we have to do it right now in the same way, because we have to know that it matters where the lower margin is defined. It matters how wide the confidence interval is. It matters how the sample size of the patients is, then, because the lower the patient sample size is, the easier it is to recruit the trial. But the higher the potential increase with the risk of a progression-free survival event is in the end in a metastatic setting, and for event-free survival in the adjuvant or neoadjuvant setting. So that means having a lower margin of minus 15%, well, it can increase the failure rate in the end. Having a rate of a lower boundary of minus 10%, this is more realistic, but you see the patient number has to be twofold higher. And having a lower margin of minus 5%, that is not feasible. So it's not realistic to recruit more than, let's say in this example, 3,700 patients for a biosimilar study. To conclude, in metastatic setting, objective response rate is the most sensitive endpoint recommended by the regulators. We have, but we have a heterogeneous patient population that is less sensitive because comorbidities, pretreatments, and changed cell clones are the fact we have to deal with. Objective response rate has to be correlated with progression-free survival, but we know that there isn't a correlation that is predictive. A 10% equivalence margin for objective response rate is realistic, 5% is not realistic. And for total PCR in neoadjuvant early breast cancer setting, we have, in comparison to metastatic setting, a more homogeneous patient population to show differences. Because they are not treated, they are healthier, they don't have these comorbidities because they're not metastatic. So therefore, we recommend to go for a study in the neoadjuvant setting with an endpoint that is defined as total PCR, and then you can correlate your PCR later on with event-free survival using a 10% equivalence margin. Potential study populations, therefore, could be in my field, in breast cancer, the neoadjuvant and adjuvant setting, the metastatic breast cancer setting, first and later line, and we'll talk about trastuzumab that is approved for gastric cancer as well, and HER2 metastatic gastric cancer. So you can decide which is your favorable patient population you want to recruit for your trial showing biosimilarity. As I said, in the early breast cancer setting, your patient population is homogeneous, and I think I don't want to repeat everything. So this is my recommendation, to go for early breast cancer, neoadjuvant, and PCR is the endpoint. And this all leads to the issue of extrapolation. Paul will talk after my talk about, because it matters if you conduct your trial in metastatic setting and then you extrapolate your data in the early breast setting from a non or less sensitive patient population to a more sensitive patient population. So I don't have any problems with extrapolation, but I want to focus on it matters from what setting you extrapolate your data in the other setting. And the other way around, from a sensitive patient population, not pre-treated, extrapolating a metastatic setting, I don't have any problems. And we always have to take under consideration that we have different mechanisms of action and immunogenicity and so on. And this is more difficult to show in the metastatic setting. So this is what I said already, just to illustrate. So it matters from what setting to you do your extrapolation. So therefore, the ESMO positioning paper says we have to have sensitive endpoints, and the sensitivity of endpoints used in the trials matters, and this is what I explained already. So I think we can skip this, and this is just to illustrate that total pathologic complete remission is a good endpoint, because we know that patients who receive pathologic complete remission, so PCR, these patients will have a survival benefit. So it's easier to correlate your endpoint with the outcome of the patients. Easier than in metastatic setting, where you have to correlate objective response rate with, in the end, your outcome. There we have data for objective response rate and the correlation with progression-free survival. Also, we don't have the data. And this is from the CT-NeoBC study that was done by Patricia Cortazar, including 12 neoadjuvant breast cancer trials. 9 trials came from Germany, because neoadjuvant treatment is the field we are experienced in. You see there is a difference whether your patient received a PCR or not regarding overall and event-free survival. And when we talk about when we talk about HER2 positive patient population, then you see the PCR with your treatment you do is more likely to generate in more aggressive patient subgroups, like here in the hormone receptor positive, HER2 negative grade 3 subgroup, and in the triple negative subgroup, and in the HER2 positive hormone receptor negative subgroup. You see, if your patient receives a PCR, and this is the reason why we do this in vivo sensitivity testing with our patients, treating them with neoadjuvant chemotherapy, you can tell your patient this advantage of PCR will be translated in an overall survival benefit. So therefore, PCR is accepted as a surrogate endpoint for development of new drugs for neoadjuvant treatment of early breast cancer center in the EMA guideline. So another issue is that we use these monoclonal antibodies quite a long time. So it's recommended, so the current use of trastuzumab in the adjuvant treatment is up to 1 year in the treatment of HER2 positive metastatic breast cancer center until disease progression, and in gastric cancer until disease progression. So you don't know how long you will treat your patient in terms of until disease progression, but in adjuvant setting, a year. So this is something you have to consider regarding interchangeability and switching. And if you have a look to these three illustrations, then we have to differ between transition studies, where the reference product goes to the biosimilar, or the patients receive the biosimilar in the end, a single switch, or multiple switches. And I don't think that we will ever have a study where patients get multiple switches, because it's too expensive in the end, and it will take too long. And how many switches? 1, 2, 3, 4, 5, 6, and what do you measure? Do you measure your safety and immunogenicity after the first, second, third, and how do you compare it? And in clinical practice, and we talked about 2 days ago, we will have multiple switches involving several products. This can theoretically occur. So we have to have a post-marketing documentation in the end to know what we do with our patients, and this is the issue of pharmacovigilance. So you can treat your patients with a reference product, biosimilar 1, and then you switch to biosimilar 2, and the reference product again. And if you have safety issues, who do I call? Do I call in terms of trastuzumab Roche all the time? Or who do I call? So this is a problem we have to take under consideration. 5 minutes left. Isn't it? We started at 20, but okay, I can end. So this is just to give you an idea that there are several studies done in phase 1. Some more in phase 1 regarding safety, and I think more interesting is what was done regarding clinical trials in different settings. So you see there is a study ABP 980, so this is an Amgen product in a neoadjuvant adjuvant setting. Then we have the BioCat and the Celltrion product in metastatic setting, and Celltrion learned the lesson and conducted a study in neoadjuvant and adjuvant. The Mylan study was done in metastatic, and so on. And the first full paper for trastuzumab, so the Heritage study, was published last year by Ho Brugo. And she showed in this study the biosimilar compound plus docetaxel or paclitaxel for 8 cycles, and compared this to the reference product. The results were similar. That means clinical endpoint was met. So no safety issues, no loss of efficacy in this study in the metastatic breast cancer patient population. And another study, and this is what I mentioned before, that you have to have a closer look to the confidence intervals, to the boundaries. This is the Celltrion study, the CTP6 in metastatic breast cancer, and the difference in the objective response rate was minus 0.05, and the equivalence margin was minus 15%. For 15%, as I said, you don't have to have that much patients. And the BioCat study, and this is not of a good quality, I have to say this was a non-inferiority trial. You see minus 20% for the non-inferiority margin, with a sample size of 110 patients in metastatic setting, and now you can understand that the companies submitted these data to the EMA were not successful. So the EMA rejected and said, no, this is not good for approval. And to end my talk with this design, so I think this trial tried to combine everything I told you in my talk, that you have a clinical trial in a neoadjuvant setting, then comparing a reference product and, of course, the biosimilar. And then you have just one switch, but you have a switch, and you go after surgery in the adjuvant situation. So this is the ABP 980 trial, and results are awaited. We will discuss them in San Antonio in the States in 2 weeks. To summarize, biosimilar approval is based on totality of evidence approach. Clinical evaluation requires careful selection of sensitive study populations, treatment naive, and endpoints. Objective response rate better, pathological complete remission. Equivalence design is the gold standard. Inferiority design is not the gold standard. Confidence interval, sample size, lower upper margin matters and can influence the study results, and due to duration of administration, switching can be an issue, and we have to document it very carefully what we do with our patients. Thanks for your attention.